The Department of Genetics at UMC Groningen exists for nearly 65 years. We were founded in 1951. And we have about 250 people working here. And we have a big emphasis on complex genetic diseases, and we really try to understand how genetic variation translates into phenotypic variation by making use not only of patient materials, but also a big population biobank, which includes at the moment 167,000 people, which we leverage with a lot of omics data. We really have the full path from gene discovery all the way to implementation in the clinic. And a nice example is preconception testing. We are developing a test where we can, before people get pregnant, can test whether they are carrying a severe mutation, both of them as a couple, and have a high chance of getting a child with a severe disease. And that is currently being implemented in clinic with the help of GPs.
I work on a project in which we try to determine the diagnosis in critically ill newborns in the neonatal intensive care units. We do this by sequencing their whole genome, and we focus on all the genes that are involved in Mendelian diseases.
Intensive treatment has a very high impact on parents and children, especially in critically ill young children. If you can use rapid whole genome sequencing, you shorten the period of uncertainty for parents, but also for the medical staff. This period is now, when we use rapid whole genome sequencing, about 2 weeks, and we are aiming to reduce that to a period of only a few days. When we have a diagnosis, medical decisions can be made, like stopping treatment or even intensifying treatment when you know that prognosis is good.
We really are kind of getting out of the comfort zone of the standard clinical genetics, and also have to work not only on the practicalities, but also on the ethical and legal aspects that are associated with that.
It's important to realize that genetics will be increasingly important for many medical disciplines, and also it will become much more complex. So many genetic risk factors to take into consideration, and also the interaction with all sorts of non-genetic environmental risk factors. It will be difficult to weigh all these different things and add them all up and make clinical decisions. We are developing several things to support that. We are building a virtual clinic for genomic medicine so that students can more easily study these complex things. And also we are developing decision support systems. First, within our pharmacogenetics program, it's important to help in real time, help the clinicians in their offices decide what the best course of action is.
Ja, genetisch onderzoek is voor mij erg van belang, omdat we een zoon hebben met een erfelijke niet te genezen huidaandoening. En dan is het wel heel erg prettig dat er genetisch onderzoek gedaan kan worden. Vooral ook met het oog op van waar zitten de fouten in het DNA. En wat heeft dat voor consequenties voor de erfelijkheid als hij gezinswens zou hebben.
Epidermolysis bullosa, it's a devastating skin disorder where patients get blisters from birth all over their body, accompanied by scarring. There are many different forms of epidermolysis bullosa, and in the most severe forms, patients die in the first 1 or 2 years of their lives. There's currently no cure for EB. So it means patients get treatment, but it's mainly symptomatic. And what we're trying to do is to remove the part of the gene by using specific RNA molecules. So we're trying to remove the part where the mutation is located, and then restore the expression of the protein that is absent normally in patient skin. We have done successful experiments in cells and in mice that show very promising results. So actually I'm very hopeful that we will be able to bring this approach, which is called exon skipping, to the clinic in a couple of years.
The main focus of my research in clinical morphology is to study children with rare chromosome disorders and with Chart syndrome, but also to study the implementation of new genetic techniques in diagnostics of children with developmental delay and congenital abnormalities. You really need to compare all these different chromosomal abnormalities and the clinical outcome to be able to predict what in a specific child can be expected. This piece of art, it was made by a child with a chromosome 6 disorder. And our project with using social media started with the Chromosome 6 Facebook group, which was a group of parents that met each other via internet. With help of this Facebook group, we really started a very successful project to collect information worldwide of children with chromosome 6 abnormalities. And within a couple of months of time, we had more information that was available in international literature.
In the last few years, thousands of genetic risk factors have been found that cause disease, but for a lot of them it's still unclear how they eventually cause disease. We currently don't know what kind of cell types are affected, what kind of pathways are disrupted. And what my group aims to do is to take a systems genetics approach that uses a lot of data that is publicly available, but also relies on Lifelines Deep, and that's a cohort of 1,500 individuals for whom we have in the last few years collected a lot of different molecular information, including gene expression, methylation, microbiome, metabolite data. Right now there's a real explosion of very interesting big data, and it's this unique combination of data generation and also the development of novel tools, novel methods to accurately analyze this data permits us to answer very interesting medical questions. In our department we have a lot of different people from many different nationalities and a lot of different disciplines, and that really helps to think about problems in a different way. We don't have any offices here. Everybody's working at a flexible working place. The day that you are in, you are sitting next to somebody who is probably working on something completely different, and the next day it's again another person. That really helps to think about problems in different ways.
The future of our department is really to move into precision medicine. It doesn't matter anymore whether we are dealing with families or single patients, whether it's a complex genetic disease or a more Mendelian disease. We really want to use genome sequence data to help people in either their treatment or their diagnosis of disease.